how biophotonics or optical imaging spectroscopy can be used for drug discovery in big pharma. So thank you again for coming to speak.
No problem. Anytime. Great. So can everybody hear me first off? Okay, great. Actually, I was kind of wondering how this talk would fit in with an entrepreneurial type environment, or a bunch of talks on entrepreneurial activity. And actually, it fits really well, because actually at GlaxoSmithKline, I'm an entrepreneur, but I'm built into a big company. So that makes me a chicken entrepreneur. I've got a built-in customer base, and I got a built-in funding source in the company for technology development and future capabilities development. What I'm going to talk about today, though, is the potential role of biophotonics in the pharmaceutical industry. And I put potential in here because pharmaceutical industry is very, very conservative. We're controlled by a regulatory body, and changes that we make to our processes can impact our long-term drug development process and cost us tons of money. So whenever you look at biophotonics, which is a relatively new field, especially whenever you're looking at it from the perspective of pharmaceutics, where we typically develop things on 20-year time frames, this is a leap of faith, or there's a lot of things we can do with it, but we haven't done yet. We're doing some of it. That's my particular role. Like I said, I'm an entrepreneur within the company. My job is to try to go out and find these techniques and capabilities, bring them in and implement them into GSK if we see value in our proposition. Talk outline today. So today I'm not going to talk necessarily specifically about what GSK is doing with this. A lot of this talk is really related to the general space of where biophotonics in particular can have an effect on the pharmaceutical development process. I will talk a little about GSK. I work for them. Hopefully you can get a perspective of how big we are and what we do, etc. Give a little bit idea of the breadth of the different capability or different types of drugs that we're developing, space that we operate in. The next thing that I'll introduce is the drug development process, which is key. So if you're developing or you're an entrepreneur and you want to go out and develop the next good product, you better understand the process that you're implementing it into, where the shortfalls are in it, where the money is, how much it's costing to do things, how long it takes, etc. Because whenever you're developing that value proposition, you got to be able to hit the bottom line. Then I'm going to jump into some clinical applications. I'm actually going to give you a cross-section of where biophotonics could potentially play a role in GSK and in the broader pharmaceutical environment. One aspect of this, so whenever you think of biophotonics, typically people think of biophotonics as a clinical diagnostic tool. GSK, on the other hand, is not necessarily interested in the clinical diagnostic aspect of this. I'm really interested in can I see my drug have an effect on that disease, not on the disease itself necessarily. I'll take a tool that sees the disease and can be utilized for a drug, but that's not necessarily my focus whenever I'm looking at these things. We're going to talk about formulation detection. You'll understand a little bit more about why we'd be interested in that, discovery and the drug detection, commercial support, biophotonics outside the body, because a lot of the biophotonic tools that you utilize and develop can actually be utilized in other areas. We'll end with some conclusions. So as I said, I'll start with a little about GSK. This is our standard slide introducing the company. Our motto is do more, feel better, live longer. We really try to live by that. We are controlled by a bunch of values that are at the top of our list. I know that we've been in the media for some rather bad things in China, like bribery. That is not by any means what the company is out to do or the values by which we live by. I can tell you people are getting fired over that today, and we are going through tons of training to correct any problems that we have in that particular area. We're really patient focused. Everything we do is really around the patient, making sure that we're transparent in what we do. We are the only company that is currently publishing all clinical trial results, or at least giving access to anybody who needs access to all our clinical trial results, and we really strive for integrity. Our strategic priorities: grow a diversified global business. You'll see that we are quite diversified in what we do. We actually have portfolios in the pharmaceutical world where you get drugs. We have commercial space, and we're also huge in vaccines. We want to deliver more products of value. So instead of going for the Me Too drug like a new Viagra, we're actually going after other things like Alzheimer's disease, or other disease areas that are not well established or don't have established drugs to be utilized in those spaces. We're trying to simplify our operating model, creating a culture of individual empowerment so that people like me who are entrepreneurs in the company can actually go out and do new and inventive things that really are game changing for the company. And then we want to build trust. Now, one thing you won't see up there is technology development. And you might be thinking, well, biophotonics clearly is not in our strategic priorities. Technology development and cutting edge capabilities is actually underlying in all this, because if we can't develop cutting edge technologies, we can't reduce our costs, we can't do better for the patient, we will not get better products to market. So it's really an underlying aspect to all that we do at GSK. Just a perspective on our size, we are about 100,000 people worldwide, 17% in the US, which is where a large portion of our research and development is done. I'm actually based out of the Philadelphia site. We also have a major research facility in RTP, our Research Triangle Park in North Carolina. We have a huge base in Europe. We are a European-owned company, UK based, and then we have a large population of people in various areas of the world. Our flagship products and pharmaceuticals, some of these you will have heard of. We are a worldwide leader in asthma treatment with our Advair discus. You actually inhale it. The discus itself actually is the more important aspect of that particular program. Valtrex for herpes, Tykerb, which I believe we just sold for breast cancer treatment. We have a huge portfolio of products that cover a very wide range of medical needs. But in addition to that, we also have a huge consumer health care line. So a lot of the time you see things that are currently prescribed by a doctor, they're on the market for a long time, there's no real safety risk associated with them. You might want to put them as an over-the-counter drug so people can self-medicate. That's actually what happened with Ally, which is a weight loss drug, Orlistat, which has been on the market for many, many years. No real safety profile with it. Some side effects, but those were minor. They took the product and made it over the counter. We also have things though like toothpaste and Panadol for headaches, which is really a UK product, Lucozade, just a broad portfolio of different things that we have for consumer healthcare. On the vaccine side, I don't have a nice slide like this for two reasons. One, I didn't have one that I could easily repurpose. And two, I've not done any work in the vaccine, so I'm not really going to talk about that today in this. We have a huge portfolio of compounds and different products that we put out there. The real question, or the real thing that you really need to understand, is the drug product development cycle. Because that's key. If we understand that, you can then start seeing where you can potentially apply biophotonics. And actually, part of the purpose of the talk today is really to get you to think about where biophotonics can play a role in this. I mean, I've thought about this, and you'll see it up here, but new ideas are what we're really trying to generate with this program, challenging the old way of the way we do business. Typical drug development process, we go through discovery, which is our first stage, where we actually have to find a target. We have to find something we can inhibit, activate, block, do something to with one of our drugs that's going to change the biology. MEK inhibitor. We're inhibiting the MEK pathway so that the cancer doesn't go. BRAF inhibitor, same thing. We're going to inhibit a BRAF pathway so that the cancer doesn't grow. You have to understand your target. Then you have to develop a drug that actually hits that target. Once you have that, then you can actually start going on from cell-based type stuff, where this is actually the cheaper end of this, and I'll show actually in just a second the important aspect, the cost and timelines associated with these processes, into the more expensive, longer time frame activities, which get you into preclinical development. Preclinical development, this is where we start doing our safety studies. We start scaling up the development of our drugs from microgram type quantities all the way up to kilogram type quantities. So there's a huge scale up, a lot of cost associated with that, a lot of time. It's not easy really to take a little beaker of stuff and actually make it into big beakers, believe it or not. Huge cost, huge timelines associated with getting these drugs from the little microgram up to milligram, kilogram type quantities. And we start the process actually making the drug, what you're ultimately going to see and take in the clinic. We're doing our safety studies at this point in time, which is what our little animal is up here for, to show that our drug is safe and that we're not going to hurt somebody with it. And you might also be doing some efficacy studies to see if you actually have an effect in the animal. We're going to take that, we're going to put it in the clinic and start doing our clinical development, start looking to see, one, is it safe for people, phase 1 type trials. phase 2 type, where we're going to start seeing is it safe and efficacious in patients. Phase 3, where we do big studies to make sure it's really safe and that we see true efficacy in our patients before we actually go out and we launch our drug. As I said earlier, though, as an entrepreneur, you really got to follow the money. Well, the gentleman who presented just before me said, follow the money. That's actually what we have to do in order to improve our drug development process. First 3 stages of our drug development process are shown here. And this is actually out of a 2010 paper by Eli Lilly that all pharmaceutical companies use as kind of a broad guideline for the typical cost of a drug and what it takes, timelines, and percent of probability of technical success, our cycle times, number of projects needed for one launch, out-of-pocket costs, and our capitalized costs. These are actually the key metrics by which we have to measure whether we're having an effect on overall bottom line at GSK. And these are the key metrics by which we have to charge back to the customer. So you look at this is our first 3 stages of our discovery, or the first 3 stages of the development process. This all falls under discovery. You have your target validation, which is where we're looking for our target, trying to find what we're going to hit. You have a lead discovery where we're doing a high throughput screen on that target, trying to figure out, do we have a drug someplace that'll actually hit it and inhibit, or do something that's positive in the chemistry. And then we try to optimize that drug to make sure that we've got a class of compounds or something that actually move on to the next stage where we're actually going to test it for safety. Look up here, you see probability of technical success, 80% on target validation. I can typically find a target. If I find that target, I know that it's an active target through the science that we're doing at a cell-based level. Lead discovery, we go through a high throughput screen, we put our drugs in there. You can see very physical evidence. There's testing that can be done to show with a high degree of probability that you're having an effect. Optimizing it, I've had an effect in my high throughput screen. I'm likely going to have an effect whenever I actually optimize those drugs for it. Cycle time, 1 year for target validation, 1.5 for lead discovery, 2 from lead optimization. Understand that we typically are patenting at this point in our cycle. So this is an important aspect of the drug development cycle. For success, so if I'm going to have one drug go through, I have to have 24 drugs start off in the target validation stage. I'm going to trit out to 19.4 in lead, 14.6 in lead optimization. Out-of-pocket costs, so if I were to just go out and pay for this, if I had an unlimited piggy bank in the backyard, it's going to cost me $24 million on average in our target validation stage. Lead discovery, $49 million. $146 million to start the lead optimization aspects of this. Now this last number is a little tricky. The capitalized cost per launch. This is actually the real dollar cost to a company. Because we actually, if I put $1 in on day 1 of target validation, as an investor, I want so much money back out of that. We typically calculate it out at 11% per year. So it's capitalized over the time period from day 1 to drug end, which means that these numbers at the bottom are what it's really costing us to make the drug, because we're putting this money in, we're actually borrowing it from investors because we don't have a big piggy bank. And then we have to pay out in dividends, etc. We have to make that kind of money back in order to actually get a dime off this drug. And
Are those costs in each phase cumulative, or is that the cost for that phase?
That is the cost for that phase. You'll see cumulative in a minute. So we are talking $94 million target validation, 166 for lead discovery, and 414 cumulative across our development cycle. Well, that's, I invest $1 on that day. I'm going to ride it all the way through to the actual launch of the drug. That's how much I'd have to have. That's actually the cheaper side of the process. We get into preclinical evaluation, only a 69% chance that we're going to get the drug through. We got to have 12.4 go in. It's going to take us a minimal of a year to get that through the preclinical, where we're doing all our safety studies. We're scaling up our drugs. We're trying to develop that formulation. Out-of-pocket capital is going to be about $62 million, $150 million that we're going to have to pay back to our stockholders. Now we get into the really expensive stuff where we actually start putting this in the clinic. We go in for our phase 1 trial, which are typically smaller trials to see in healthy humans in most instances, but in drug development for cancer, this may actually be in cancer patients. You're looking at 54% chance that it's going to fail, 1.5 years of cycle time, so 1.5 years of development. 8.6 projects have to go into that to get one out, 128 million in out-of-pocket cash, 273 million to actually fund that. Phase 2, 319 million, 2.5 years of development, 4.6 drugs go in, only one's going to make it to the end. Another 2.5 years of development time, 34% chance that it's going to fail at this stage. Typically, phase 3, you get a higher probability of success because you normally attrit out at phase 2, but you still have a 70% chance or likelihood that you're going to have this drug fail. 2.5 years more cycle time. 1.6 projects have to go in because of higher likelihood. 235 million out-of-pocket cash, 314 million in actual cash. And then registration and launch, which is actually cheap because once you've got through all your phase 3, you got all your data, you know that this has a high probability of success, you know what the safety profile looks like. High probability it's going to get registered and launched, although you're always at the whims of the FDA. Ask MannKind, they've gone 3 times with their Afrezza drug, and it looks like this time they may get their approval. Out-of-pocket cash, only about 44 million, 48 million total. Looking at these numbers, you can quickly see where the big numbers are at the bottom. We have lead optimization at 414 million payout, 273 million in our phase 1, 319 in our phase 2, 314 in our phase 3. Just to give a perspective of overall totals, only an 8% chance that your drug is going to launch. So 8 out of 100 drugs are actually going to make it out the door and actually go into the human. As a registered product, we have a 13.5-year cycle time on a 20-year patent. We have one project that gets launched, 873 million in out-of-pocket cash, 1.8 billion to make that drug. So whenever you're hearing about high prices of drugs, etc., that's why. $1.8 billion went in to make that drug. We have to return all that capital, plus we have to make enough to actually invest in our next round. So it's a huge cost to make any particular drug, and especially for drugs that are less, that have less of a population out there who need them. You know, specialized drugs for specialized situations are very expensive to make because of this particular cost. That's part of the goal of my program, is actually to bring that cost down. What are our biggest risks? Biggest risk is one word: attrition. If you talk to anybody in the pharmaceutical market, they will say attrition, number one risk to any drug. Because if that drug attrits out at phase 3, I've just lost almost $1.8 billion. Phase 2, I've lost, 1.4 billion. It's a huge cost to us if a drug, as we move along its development timeline, to have a drug suddenly fail. Why do they fail? We'd have no efficacy. Drug goes in, nothing happens. We do all these models. So animal models, we're trying to get away from them, actually. We don't like using animals. We have to use them for safety because I'm not putting a drug in a human without having the prior knowledge that this is a safe drug for you and not going to kill you, not going to cause liver, heart, etc., or at least have a high probability that that's not going to happen. On the other hand, efficacy models in animals rarely work. So there's no reason to really do them. We get them into human and you just don't see anything. Even though all of our discovery aspects, where we're doing cell-based assays, etc., show that there should be an effect. Second issue, unsafe. You get it in a human and for some reason it hits a target outside of what you're expecting. So for example, I'm targeting the heart. I got a drug that I'm taking systemically, so I pop in a tablet, it goes through my digestive tract, it goes right into your bloodstream, hits your liver, your lungs, your heart, it hits everything, and suddenly there's some metabolite or something in some other system that we're unaware of that suddenly cause issues, and suddenly you see a safety hit. Testicular problems, diarrhea, stomach bleeding. You can list off a bunch of different things you can particularly get. These are typically side effects, but if the drug's unsafe, we can't actually market it. The last one actually is related to the efficacy aspect, which is we can't get to the site of action. So drug goes in, but drug doesn't get to the area that you're interested in, such as like prostate cancer. Prostate cancer is very well protected. It's very hard to get a drug to the prostate cancer. We got lots of drugs that seem to work in smaller models, but don't seem to work in that particular aspect. Next biggest risk is our drug development timelines. 13.5 years to develop a drug, 20 years patent life. That leaves us 6.5 years on average to return all that money, that $1.8 billion, which, you know, do the math there, that's $300 million a year minimal that you have to return on that timeline to actually make a dime off the drug. And we are trying to, you know, continue our pipelines and continue making the next generation drug that's even better than that. So, next aspect of this is related to efficacy and why you have problems with efficacy. Now, I've made these numbers up for demonstration, but they are actually what you typically see in a clinical trial. So in a clinical trial, I'm looking at the patient response with regard to the efficacy. We have some placebo that we're going to put in, and we're going to see essentially nothing, right? Placebo, sugar pills don't cure cancer. Although you see people who have response to cancer for sugar pills. This is what I'd like to have. I'd love to have this, where we see day one, we give the drug, and suddenly it goes off, and you know, we see an effect, and you can clearly monitor it. There's systemic or some marker there that I can actually mark and see real positive results. Very unlikely. We don't see that normally in our trials. I'd love to have the little boxes here where we're seeing some kind of linear response, where, you know, just seeing gradual improvement over time. Again, typically doesn't happen. More likely to happen is something like this, where we actually see some very, very, very slow response that then we see a response curve over time. And I actually haven't shown the other aspect of this, like in cancer therapy, where you actually see this followed by this because the cancer cells adapt to what we're treating. One thing in this graph, though, that you should notice is I put a timeline down here with no numbers, and there's a reason behind that. Our problem with efficacy is often the fact that we can't look across this entire timeline. The development of any disease typically takes lots of time. We're typically looking in this region here, a couple of years. That's the only, you know, if we had to go 6 years for a clinical trial, we're outside our patent life. There is no viability for our patent, no viability for the product. We can't make any money off of it. Which is unfortunate because we are a drug development company and we're not privately, or we're not funded by the government, so we have to make money off it. What you typically see in that region is something like this, though. So this is actually representative of what you would see in a clinical trial. We have our placebo in diamond, and we have our drug actually here in square. Is there a real response there? Now, I know there's a response because I actually put this together, and I know there's a factor of 2 response to the drug because I actually modeled it on purpose to have a response. This is what you often see whenever you're actually doing the clinical trials. If you look at drug efficacy versus placebo, you will see placebo kind of bounce around and give you some response in some patients. Some it won't do anything. You'll see your drug bounce around. What causes this? Long duration of the disease progression. So it takes a long time for the drug to actually have activity because you're waiting on something to change in the body that slowly shows up in the systemic or the methods that you're measuring. You have a lack of direct measurements of the drug's effects, and this is actually where biophotonics has a huge potential within pharmaceutics. You have subjective measurement by a doctor or nurse. I'm going to look at it and see, does it change, you know, visible aspects of what's going on. You're going to have indirect measurement using surrogates to determine what's going on, what the effect is. So I pull your blood and I'm looking at a drug that's systemic. I'm not looking at the actual disease and the actual disease progression. I'm looking at markers for that disease. So there's a chance that I'm affecting one aspect of the disease, but another pathway is ramped up and you're, you know, running off. Biological variability. So everybody's different. One patient responds to sugar pills, one patient doesn't. These are all factors in the noise aspect of this. Give a what's that?
Yes, how much of personalized medicine is being considered in the big pharma?
We're considering, but it's cost. It's expensive because each person's different. So if I'm making a drug for one particular phenotype, actually MEK inhibitors, BRAF inhibitors, those are actually personalized drugs. If you have metastatic melanoma and you are not BRAF positive, you can't take a BRAF inhibitor. Same thing with neck. So we're going more and more towards that because the targets are better and get rid of some of the biological variability. But it's a very expensive path to go down unless we can reduce our timelines and costs.
When that expires, by how much do you offer? People still buy the drug, right?
No. Well, they buy the drug, but typically patent expiration is followed the next day by generic competition.
And then what would you expect sales driven?
Oh, easily. Ask Pfizer right now. Viagra and Lipitor just went off patent. They lost from I think Lipitor was a $25 billion a year product. I think it's down to expectations 3, maybe. And within a few years, it'll be down in the millions easy. I mean, generic competition is a huge factor in this because once your patent expires, all the generic companies now can manufacture your drug, and actually their cost to actually bring to market is significantly lower because they don't have to go through all the safety aspects. All they have to actually do is show that they have an API that's the same as your API in a formulation similar to your formulation that whenever given to people has similar effect. Now you can talk to people, and actually, you know, it's an interesting conundrum with generics because some generics are actually better than the original. Some generics aren't. You don't necessarily understand it because the generic is all the same API, but there's placebo aspects to it. You know, add caffeine to Excedrin. Now, granted, caffeine is considered a drug at this point, but caffeine with acetaminophen has rapid effects, which improves actually. That's why Excedrin works so quickly, whereas just straight Tylenol doesn't. So, you know, you have those effects, but they don't have to go through the development timelines that we do because they don't have to discover target, drug, any of that. They start essentially at phase 3. They already know it's safe. They already know it's efficacious. All they have to do is put it in a formulation, tablet it, put it in people, show that they have a comparison study that's relatively same. Out the door it goes, and typically the day after your patent expires, you have a generic competition immediately because they can do all that beforehand. Going to the long duration of disease progression, though, which is what's shown up here is the progression of geographic atrophy. Geographic atrophy in ophthalmology is an interesting area for biophotonics because whenever we treat geographic atrophy, there is no way to systemically tell whether our drug is at the site of action. It's all in the eye. It's all in the retina. I can't pull your eye out and do any tests. If you'll volunteer, we'll take it. But how then do we tell that we're actually having an effect on the patient? The only way to tell is the patient actually goes in. They can do some imaging like this, which shows you, I mean, you can clearly see that the geographic atrophy is progressing across this patient. But the real indicator for geographic atrophy is your visual acuity. If I can still sit and look and got 20/20 vision, I can have all the geographic atrophy in the world. It doesn't matter. How can I measure that I'm having an effect on this person's geographic atrophy? To give a perspective, this is a 29-month time period that's shown up here. The actual growth profile of geographic atrophy is on the order of sub-micron or right at micron level per month. So if I'm doing a clinical trial around an ophthalmology drug, it takes a long time for that drug, for me to determine that that drug's having any form of effect. And it's very hard for us to determine that whenever we're doing that, it's very hard to have your attrition risk becomes much larger because I can't detect whether I'm having a systemic effect. I can't detect very easily what the effects are early on, which means that my trials have to be longer, my costs go up. This is why you don't see a lot of drugs in the ophthalmology space right now. And a lot of companies aren't working in the ophthalmology space because of long timelines and the lack of capability to actually see what's going on. So let's talk a little bit about clinical needs. So we're going to break down now into actual, potential areas. Now these are mainly focused on skin because that's an easy mark. It's low-hanging fruit for the most part. And just to give perspective, we actually do use biophotonics. I mean, we use a variety of different imaging modalities in our studies, but for the most part, these are old school type technologies. They've been around forever. They're well established. They give us information that we're very interested in, but some of them are only related to animals, so we can't actually use them in human. We prefer things that translate into the human. Some of them, don't tell us some information about what's going on, but aren't specific enough at the cellular level to tell us really what's going on. So we can, utilize these to give us some information, but we really need to know in drugs whether we're making an effect at the chemical level, at the cellular level. A lot of these techniques are not around that. So that's really where we're seeing the real need for biophotonics, high resolution, deep penetration, capabilities to image where our drug is at its site of action, and what's it doing there. If I had my ultimate system, so for you guys, since I've heard you talking about entrepreneurs, it's great. I want y'all to build me a tricorder because it's handheld, non-invasive, it doesn't require any contrast agents, no patient prep. I can walk up and scan you and I know whether you're disease. It was capable of being used across species, so we could use it in our animals, in our cells, everything else. Likely detected events at the cellular molecular level, because that's the only way they could have done some of the things they were doing with it, and it could penetrate the whole body. This is what I want. So if you're an entrepreneur here today and you can build me this, we will pay you big money. Reality is we know we're not going to get that anytime soon. But what we really need to do is move our clinical evaluations from quantitative or qualitative to quantitative. And as I said earlier, the difference between pharmaceutics and typical medical devices is the fact that I'm not necessarily in need of diagnosing that this person has a mole with melanoma or a punch biopsy to determine whether this person has melanoma. I can do that with other methods. I need to know at an earlier time point, at a higher resolution, is my drug having an effect on that? Because then if I can do that, I can reduce my clinical trial timelines. I can reduce the size of my clinical trials. I can attrit out drugs at an earlier time frame, and I can drop that $1.8 billion down quite significantly. We need to actually move this over to where we're actually seeing this in vivo. So this is actually some data from GenLab showing, a in vivo biopsy, optical biopsy with one of their systems, looking for melanoma and showing the high degree of contrast that we can get out of this, the different aspects that we can see. We can see molecular level detail. We're seeing all kinds of stuff that traditionally you'd have to take the punch biopsy, stick it under a scope, do a bunch of different tests to see. If I can take that and start applying it to some of our drugs in the dermal space, that's a huge benefit for GSK. So I'm going to use actually an example of some work that we've been doing with Professor Beaupart here at the university. And we're going to be looking at the effects of topical steroids. So what are some of the reasons that we might want to look at this? One of the risk factors for topical steroids is skin thinning with long-term use. You stick it on the skin for long enough, you will see your skin thin out. You will over time actually see wounds uphill occur. It becomes easier to tear, etc. The mechanism for thinning is not well understood. So whenever we do these studies in the clinic, it takes a long time to actually do. In our traditional evaluation was mainly subjective evaluation by a doctor who did look and feel this. It's, pinch the skin, see how it feels, does it feel better? That's how we would normally evaluate it. I can't tell you what steroids we used. GSK made me pull out the steroid names yesterday, even though we don't sell them, but they want it published before we actually reveal that. Purpose of our study that we're going to show here was to perform in vivo longitudinal study of changes that occur in skin with topical steroid treatment. Now, this is done in mice, so we're actually two benefits out of this. One, we can monitor these animals longitudinally. Normally we'd have to put them down, so we'd have to use more animals. Second aspect is we're actually monitoring the same animal over time, which reduces our biological variability and allows us to see the changes in vivo. Treatment. Couple of different steroids twice a day, 2 mg per square centimeter as possible endpoints. Change in skin thickness, changes in metabolic rate, morphological detail, potentially even detection of drug. All studies were conducted with IACUC approval, so these were done according to the legal requirements. So what we want to get to, though, is something like this. And I can thank Andrew, who's in the room, for these very beautiful images that we have of the skin. traditionally, the way we would have evaluated these mice is you would pull the skin up, look, felt them, see if they start to feel papery, pull them apart. Here we can actually see lots of detail in these. You know, you can see the layers of the skin, you can see the hair follicles in the skin. We cut out an area here, my image is not working. You can actually see the keratinocytes in this skin. We can see a lot of detail. This is detail that we would traditionally have to take a punch biopsy and pull out and then have a look under a microscope. In this case, we're doing it in vivo. You can see metabolic changes with this treatment. So we can see on day 0, day 1 are controls, day 21 are controls. You can see the lifetimes there. Lifetimes of the control animals are fairly tight. We get into the steroid 1, by day 21 we're starting to see changes, we're starting to see increases in metabolic activity. Steroid 2, we're starting to see metabolic activity at day 14. So you know, you can see in vivo with these, the changes that are occurring due to our drug. This is the type of information that we need for our studies in GSK. We develop a lot of dermal compounds, so this is critical to us. Traditionally, again, I'd have to take a punch biopsy. How many punch biopsies can you take on a patient who has psoriasis? Maybe one, maybe. So traditionally, I just have to wait and watch and see whether or not I have effect. Looking again at collagen, you can see, you know, the high level of detail that we can see in the collagen level. So we're seeing changes, you know, control animals, nice spindly fibers, you know, well separated. But you start looking at our treated samples with the steroids, again, looking at this in vivo longitudinally, and you can see that this animal's changing over time on steroid 1. You can see changes clearly in steroid 2 being applied. Again, how many times can I look at that with psoriasis or atopic dermatitis, seeing the actual effect of this drug on the subcellular aspects of this? In human, very little activity to do it. So the purpose of showing those is actually to get to the point of what we really want to do. So atopic dermatitis, psoriasis, things that we actually are treating, but we can't actually take a punch biopsy. We have to depend on visual acuity or, you know, doctors to actually look at. We want to be able to take these types of technologies and techniques and see directly what my drug is doing there. So, you know, what we want is new clinical endpoints with earlier endpoints, reduce clinical trial sizes and timelines, as I said earlier. Which reduces our time to market and overall cost of the drug. And then it also improves the patient experience. And without the need to do punch biopsies, you know, that patient can come in, we can evaluate them on a much regular basis, actually give an idea of whether our drug's really having an effect. You know, a lot of studies have what's called rescue treatments. There may be a treatment that you have out there that's already improved in the market. You're trying to improve on that treatment. If I have to watch you for 24 months before I know there's an effect of my drug, that means that you're off your normal treatment for 24 months before you go back on it. And if I had no effect, you know, I may not have done any harm, but you might have been better off on your other drug. If I can know that earlier, I can rescue you earlier.
So a lot of the imaging or analysis is to look at efficacy. But how much, I guess, of interest would there be in understanding the mechanisms of the disease? And therefore,
About there. This is just the clinical side. We'll go on to formulation, then mechanistic. So next aspect of this is we have to develop a formulation. So whenever you apply a dermal drug, and again, I'm going to stick to the dermal space, one of the aspects that you have to ask yourself is did my drug ever get to the site of action? So did it actually penetrate the skin, for example? So you put on a lotion of any kind, you go, okay, this is going to have an effect. Not necessarily. It's really dependent upon did you actually transmit across the stratum corneum, which is an organ actually designed to keep things out. So getting across it is actually not as easy as it seems. I'm going to show up here actually a study done with Raman spectroscopy showing again some biophotonics in use to actually track transretinal transmission through the skin. We have our Raman spectrum here of the skin. We have it at different depths, up to 10 microns. That's actually a limitation of Raman, is that we can't get the deep penetration depths that you can with other techniques like what Steve's developing. We see our transretinal peak right here in the middle. Nice, pretty peak. It's very separate. It's very distinct. Now let's look at this, though, from a formulation perspective. I take that transretinal, I put it in 3 different formulations. All these formulations are standard formulations that you utilize in typical dermal development. If I were to do this study with 1% Triton alone, and we'll focus on the normalized data over here because that actually takes into account background. If I were to do it, you see no penetration at 7 microns essentially, or very, very little. So essentially, this drug would probably fail. I would see no response. I go down here to 1% oleic acid. About 4 hours later, I might see a little drug actually make it through. PG and ethanol, on the other hand, 1 hour later, we've got drug coming down. It's actually transmitted across the stratum corneum. Which of these 3 formulations would I like to develop as a formulator at GSK? Clearly, I need this formulation. Without the capabilities of biophotonics, though, we don't have the capability to actually see that. We'd be dependent upon just monitoring what's happening with the disease and what's going on over time. What we'd really like to do, though, is more than that, because just seeing it transmit through the body tells us only did it actually transmit through the skin. What I really want is to see imaging of it. I want to see where did it transmit across the skin, what pathways did it follow. Your skin is actually full of holes. It's got hair follicles, sweat glands. You know, if you've got an abrasion of some kind, you can get drug through it. This is actually a study done by Sunny Z showing deuterated PEG, which is our formulation carrier, and ketamine. And they're following ketamine, this compound here, and looking at the transmission of it over 134 minutes through the skin, showing, you know, that essentially the deuterated PEG, and they deuterated the PEG so they could clearly see it, ketamine has a nice, pretty spectrum that can be differentiated from the skin, much like transretinol. And you can see where you see the deuterated PEG in the skin, you can also see our drug showing up there. So this is critical information, though. Knowing how that drug transmits through the skin can tell you whether you're targeting the right thing, whether it's getting to the cells that you're interested in again. And actually seeing that visible image is really seeing is believing. You know, you can really make a call on this. And actually, in formulation and drugs, etc., we don't want to tag these to do this. You can do some of this with tagging, but as soon as you tag it, you've got a new molecule. It's not the same thing. We'll talk about flipping this over, though. So let's say that I've got a drug that I want to get to a site and actually activate it there instead of actually just seeing it. We want to actually look at, this is an area that I think is really cool. GSK's not doing much in it right now. But could I actually externally activate that drug? Could I actually put it in something, send it to the site of action, have it in some way, so for example in this little image here that I still have it tagged in a way where it actually tags onto something in the body and then hit it with a light wave and then release all my payload. Why is this so critical? Well, one of the things I said earlier is often one of the things that we see in drug development is when you develop the drug, it's efficacious. It might be gangbusters on what you're actually trying to treat, but it also kills your liver. That's not a good thing. Not what I want whenever I'm developing a drug. If I can utilize this, though, now I can target it to the particular entity of the body that I'm interested in, because I'm only going to activate at that site. So if I'm interested in the heart, I shoot my light wave into the heart, activates the drug only there. I don't have my safety issues with the liver, and I can actually deliver a larger payload. So for like cancer treatment, where when you're delivering systemically, you have to be very careful because too much drug kills all your cells, just like chemotherapy. In this case, I might be able to load it up with the drug, and if I'm only delivering it to the sites that I'm interested in, I can actually activate it there and have a huge payload effect. Just another area where formulation can pay effect on this and where biophotonics can be actually utilized for formulation development. I'm going to try to hurry up. I know it's 12:30, everybody's hungry. Drugs at site of action. So this gets into the mechanistic side that Steve was asking about. One of the key aspects of drug development is, did it actually get there at the molecular level? We know where the target is. We did that in discovery. We know we got a drug that actually attacks that chemistry, can make an effect. We put it in the cell and we see some effect, or we don't see some effect. Did it actually get there? That's often the case. You know, what we'll do is we'll see some kind of effect in the cell culture. As you typically will take those cells, wash them, lyse them, take the cytoplasm, measure it out, and see if there's any drug in it, and then you assume that the drug made it intracellularly. There is no real good way of measuring whether or not a drug is intracellularly outside of that. So a drug that's tightly bound to the outside surface of this cell that could be having some effects, but very, very low quantities in the cytoplasm where I want it, would still show up in our analysis. We would still think it's making it to the site of action. And this is, again, some work that's been done here at Professor Bopart's lab, where we're trying to actually tackle this problem with biophotonics. We have one spectrum here that's taken in the background. I've kind of taken an area out of this. I don't think it was that particular wavelength, but it was in the background. You can see where our cells are. You can see some background signal. We took a spectrum where the drug should be the only thing actually transmitting, or in this case, scattering, which is shown in this diagram. Ratio of the two, and you can now see where our drug is intracellularly. This is key to us because we now know that that drug is actually intracellular, that it's compartmentalized someplace in that. And I'd love to take this one step further and say, I know not only where it is in that cell, which compartment is it in? Is it in the not macrophages, the various compartments of the drug? The organelles, the nucleus, the chromosome, wherever that is, that's where I want to see it. I want to know exactly where it is so I know whether it have an effect where we need to have it. You can already do this, or you can also translate this over into molecular histology. So traditionally, these are histology slides of testes. Traditionally, a histologist is going to take this slide, he's going to look at it under a scope, they're going to stain it in various ways and try to figure out what's going on at a molecular level. I'd like to take these technologies and actually utilize them for molecular histology, where we can actually take those same slides and get a whole lot more information without the staining. Actually see where our drug is, see where the molecular changes are occurring, where the metabolic changes are occurring, morphologic changes, all in the same system. Really improve our capabilities to utilize histology. So this is actually an area we're really interested in, where we're improving molecular or histology capabilities. So I've talked a lot about our aspects of pharmaceutics and where we can use this. Now we'll get into the commercial side of this, and just a couple of slides on it. I'll go real quick through these. There's actually a whole money side of this, which is in the consumer side, just making claims. Do we know what's going on? So I got two pictures up here. Whose teeth are whiter? Anybody? How do you think they tested which teeth are whiter? Come on. Bingo. It's entirely subjective. There is no measurement for your teeth whitening. They might take a photograph and they might try to do something where they put a little white strip against it. Entirely subjective. Most commercial claims on commercial over-the-counter products are based purely on subjective observation. So 9 out of 10 dentists said that they use Colgate. All the Colgate representatives go in and give them Colgate, and they give it out at their offices for free. Of course, a 9 out of 10. The 10th one actually was Sensodyne. So that's how a lot of your observations are made, or how a lot of your claims are made. There are no medical diagnostics out there for the test of most consumer conditions. How white are my teeth? How dry is my skin? You go to a dermatologist, you're going to say your skin is dry, go get some lotion. So I challenge you to go to your local pharmaceutical company or pharmacy and have a look at all your different options for lotions and tell me which one's better. I have no idea which one's better, because quite honestly, what they use is a metric. I mean, that there's no real test out there for this stuff. Now, I'll give you a couple of the advertising things. Vaseline jelly works by creating a sealing barrier between cells and dry or damaged skin, which locks in moisture and speeds up your skin's natural recovery process, helping it to heal from within. Half of those statements can't be supported because there's no test for it. I mean, they stuck it on some people. The people said, oh, I feel a lot better. And then they assume, based off of what they know about the chemistry of that particular substance, as to what it's doing. Same thing with Eucerin. Smoothing, repair, and professional repair secret weapon is a unique, groundbreaking technology that combines skin's own natural moisturizers with 2 innovative ingredients: ceramide, 3, a component of healthy skin barrier, and glucoglycerol, a novel moisturizer. Okay, fair enough. Ceramide 3 is part of your natural skin barrier. Can you prove to me that they've actually got through the stratum corneum? Or are you just sticking it on top? I don't know. has an effect, and there's only, I've shown a few here. I actually have pictures of whole aisles of lotions that are out there on the market today. This is an area where biophotonics can really make an improvement, because if we can actually see an effect from our lotion, we can actually then go out and market it off real science and say, we not only have the capability to do this, the people who are utilizing it like it and feel like the feel, etc. Here's the science behind it, and this is why it's working. And it also helps us define the next generation of products. So I'm going to just blow through these, expanding on these applications. So I've talked a lot about biophotonics in the body. The same technologies can be utilized for a variety of different things. I came out of process analytical. I used to be in a pilot plant at GSK. Of course, I've worked across a wide variety of different technologies and applied spectroscopy. You can take the same technologies and do stuff like tablet analysis. One of the key aspects, we're seeing cracking. How thick's our coat? How much drug's there? How's the drug dispersed within that tablet? All key aspects of the drug development process, but it's all outside the body before it ever gets in. Same thing using this for, something simpler like near infrared for loss on drying. You can use some of the same technologies you're developing for biophotonics and expand into other areas and other avenues. So last slide, GSK is my operating model. I was going to go in here and actually just circle out things that GSK finds benefit in from the biophotonics space and photonics in general. But the reality is that GSK as a company can find huge benefit in this. It's actually how I make my money, how I work as an entrepreneur in GSK is finding the different avenues, different customer bases, and different potential applications for these technologies as we move into the future. Expanding our capabilities so that we are at the front cutting edge of technology so that we're reducing that cost, reducing our clinical trial timelines, reducing our cost to consumer, and having a better patient experience. Conclusions. I think you get the point. The pharmaceutical development process is long, costly. Many areas of the process can benefit from technology. I've shown you a cross-section today. I'm sure there are many other examples. And I'm sure some of you have thoughts on where that can be used. Methods can be expanded into other space, and it really can, biophotonics can really make a huge impact. So, last ones. Your questions either equal my answers or me wanting to run for the door. So with that, I'll end. I'm sorry I ran over, but I think I started late too. So with that, I'll take any questions. Yep.
You bet. Very interesting talk. So business goes after money. So that's what I learned from this talk. So for each drug, you roughly need like $2 billion to develop. Mm-hmm. Is that kind of contradictory to the development of personalized drug or treatment?
Well, that's what I was saying. I mean, you have to, in order to do personalized drugs, your timelines have to be faster. You have to reduce your cost in order to get into the personalized space. BRAF, MEC, whereas we normally have a 13.5-year runway before they're off, I think we developed both those drugs in 5, which puts a huge strain on the organization because you've got a ton of resource that now has to go into that, but in a 5-year time frame as opposed to a 13.5. But it is possible. But there's aspects of this that we're not in control of. Clinical trials. FDA is in control of that. I have to show them that this works. I have to have enough time and enough data, et cetera, to actually show the FDA that it works. So if I can reduce that runway, I can reduce that cost, then personalized medicine makes a lot of sense. If I can't, then we can't develop a drug for it. And I wouldn't say that we're chasing the money. We have to make money in order to actually generate a product. And we're not government funded. We get very little money from the U.S. government. We have a couple of grants for very specialized things where they really want to see a drug developed, and they know that won't happen without some help. But that's why we have to make sure that we're making our money, that we get that value back and we can pay off investors, et cetera, so they continue investing. Drug development is high risk. 8 and 100. I can go to Walmart and make 5% a year. And I'm not going to have the risk that I'm going to not get a dime out of that.
Yes. Taking your example, so who do you think would care? Like whether this
because the customer, who's not the customer, they already want to care, right? What should the company care?
Oh, no, I mean, it's all in advertising, right? If I can get it on your TV as scientific fact, most people are watching TV. It's all about advertising at that point. If I can support that claim with scientific data, I can actually put that out in my advertising. I put that out in my advertising, you will see an increase in sales because now I'm able to actually downplay their claims and upplay mine.
But the customers eventually they're not going to want to oh, because of this OCT and it works. They just want to see those simple terms on the show, right? And you can't just
that's where you have to, that's the sales part of it. That's your value proposition aspect. You're one line. That's what I get. But if I can show you an image of it working, so I show you, all I have to have is 2 images. One image of theirs, one image of mine. Showing the difference and showing that works. I'm done. I've just made, $1 million, $100 million. Just based off the fact that what people see, they believe. Yes, sir.
Are you hiring?
No. We're about to hire a postdoc. But no, most pharmaceutical companies are in a state of decline right now, mainly due to the patent cliffs. GSK hires on occasion, doesn't hire on another. I expect in 5 years we'll be hiring for roles in this area, but our uptake is quite slow because of our regulatory body that we have to also abide by. The way we utilize these earlier is the fact that I'm not making a medical diagnostic, I'm making a call on my drug.
So like something like regulatory or quality experience would definitely help?
Yeah. Not necessarily. We train that in. We don't expect people straight out of college to have regulatory and quality. But if you know it, it definitely puts you, like the gentleman said earlier, top of my list. Because now I don't have to worry about you making a boo-boo.
Yeah, so I was just wondering how GSK works with universities and research settings, the interaction in this area.
Well, we're working on one right now. It's different based off of what we need from as far as from a university setting. Professor Bopart and I are working on a very unique collaboration between GSK and the University of Illinois. Others, we may fund some research and development in the area if there's a particular cutting edge need that we need some research and development done in. We have in the past, so for example, discovery and targets or drugs. If you've got a new mechanism by which that drug's going to work and have come up with some drugs, we may just purchase you if you got an independent company, because a lot of the companies or universities will then develop their own small business, which if it looks promising, companies like GSK will buy so they can get their portfolio of drugs. But it's really dependent upon the university, the need, et cetera, as to how we work.